everyone. I'm Qingshan Wei, an associate professor from the Chemical and Biomolecular Engineering Department at NC State. And then before I move into the talks, I really would like to thank the kind invitation from the organizer to join this virtual event. So today I'm going to talk about a little bit our recent research development in the area of CRISPR diagnostics and a particular coupled technology with a smartphone-based device. So my lab at NC State basically a biosensor and a point-of-care diagnostic lab. We focus on a few technical core areas, for example, microfluidics, smartphone-based devices, nanomaterials, and then particular molecular-based assays. And then we try to integrate those technical areas to form a low-cost, field-portable, and diagnostic system can be used to address many different questions from human health monitoring, plant disease detection, environment monitoring, and also bioanalytics in the area of biomanufacturing. And today my talk will mainly focus on using CRISPR for disease detection, particularly human-related diseases. And then a lot of motivation actually come from a similar vision as several other research groups they present. One example is the cartoon you see here, published in the Journal of Science a couple years ago. Basically, what this cartoon describes is a parent, a mom, holding a portable devices which can monitor the disease conditions of his child. Not only the device gave an answer yes or no, but also the device you see is really can measure the sequence information and then send this data quickly to a remote doctor and then or pharmacy. So that's really the vision, and a lot of research people group they work on in the area of point-of-care diagnostics. And we all know point-of-care diagnostics are really referring to any medical test that can be performed outside the centralized laboratories. And then it's really our motivation try to greatly expand those diagnostics from yes or no to more molecular information we can provide. So we consider three key areas or key components needed to form a complete point-of-care diagnostic systems. It could include assay components, the chip devices, which means try to hold the assay reaction, and finally the necessary reader device to quantify the assay results. And basically our lab is working almost in all three different areas, and then we try to apply the system for many different application areas I just mentioned a little bit before, from health, human health, agricultural area, biomanufacturing, and environmental. Before we talk about the CRISPR detection, I just want to mention the smartphone-based reader a little bit. We leverage those consumer digital devices as a reading device to quantify the assay results. The reason we are interested in the smartphone as a point-of-care detection device is not only because it's a larger subscription numbers. So currently almost there's 7 billion smartphones, almost equals to the total world population. That means on average, every person in the world that they have a phone. That's a massive volume of the devices. And then also because the performance of smartphone devices it has been increased significantly over the past few years. One example is the pixel count or the image sensor that has been used in the smartphone, almost a double every 2 years. So basically the trend is very similar to the Moore's law describing the transistor density increase over the years. And not only the pixel counts has been increased significantly, but also the performance of those image sensors, which is directly related to many sensing and detection applications. So this table compares some of the example smartphone-based image sensor to those conventional benchtop image cameras that have been used for microscopes, for example, EMCCD, scientific CMOS, and you will see although the small CMOS chip on the phone, they have slightly higher noise level, slightly lower quantum efficiency, but you will see the pixel size of the smartphone image sensor basically is much smaller than the benchtop microscope sensor chips, which means they provide potentially higher spatial resolution. And then importantly, if you look at the price, there's a huge difference, orders of magnitude cheaper than the benchtop image devices. And in order to form a smartphone-based microscope, there are many different ways. And one very simple way is you can form a sort of like a two-lens system. So you can re-engineer the phone in your pocket to be a miniature as a microscope, allows you to image nanoscale or macro scale objects. So you can put many different optical components in from the built-in smartphone lens module. You can put a singular lens, you can put a ball lens, you can put objective lens, but nevertheless, they form a very simple telescope configuration. The magnification basically is determined by the focal length, the ratio of the focal length between the smartphone module and your external lens. The spatial resolution still following the optical diffraction limit, describing the Abbe equation, basically equivalent to the half length or the incident wavelengths. And if you're doing well in terms of the alignment and the choices of the external lens, recent days you can readily achieve a sub-micron resolution on a smartphone-based microscopic devices. And it provides a very tunable field of view from a few millimeters square to a few centimeters squares. And then I would say fluorescent detection modality is still the most popular detection principle for smartphone-based due to their sensitivity and specificity. Again, many research groups, they come up very elegant design of the smartphone-based fluorescence detection devices. And then one configuration we use a lot is based on very simple high-angle tilted illumination, which means you put the light source such as compact laser diode at a very high incidence angle so that a lot of the excitation light will be missed by the low NA imaging system to reduce the excitation background, autofluorescence, and the background scattering, so on and so forth. And then the image you see on this slide is our early version. A lot of the smartphone configuration is a still open design. We're using 3D printing to come up with the smartphone attachment, which you can consolidate many different optical components. And in our recent versions, the attachment will be more enclosed. So basically, you cannot see the inside components anymore. Over the years, really, we work on the smartphone devices, really try to push its analytical performance. One aspect is really interesting to see, how sensitive those low cost and portable devices could be. So sensitivity is always a parameter we try to push forward. And you will see here, over the years, we demonstrate we can using those portable devices to image single fluorescent nanoparticle, single fluorescent label viruses, and then also single DNA molecules that are stretched into linear formats. And then more recently, we start to incorporate plasmonic enhancement technology into the fluorescent detection modality. So we can really push down the detection sensitivity of those portable systems to a few fluorophores, and then eventually recently we demonstrated single molecule detection on a portable smartphone device. So the video here cannot be played, but basically this video describing the single molecule blinking, a real-time video we can capture on a smartphone device. Smartphone device can also do multicolor imaging. There are many different ways to do that. And then one strategy we apply is, again, very simple, using two different excitation sources, different wavelengths, and then using a dual band filter to enable two-color imaging on the same platform. And using this device, we demonstrate one application is try to detect the cancer mutation. So one of the most common cancer mutation is a KRAS mutation, which is a point mutation, very common in many different type of cancers, and we together with a collaborator, we developed a molecular assay technology based on rolling circle amplification, which you can differentiate wild type and the mutants sequences using 2 different fluorescent colors, and finally using a smartphone to capture the 2 different fluorescence labeling. So we first test the technology in a fixed cell line. We perform in situ amplification in the cells, and using the cell phone to visualize those green labels, and then finally apply the technology to a real patient samples. So here is a patient pathology slide, and then we do in situ staining, and then use a smartphone to scan and try to detect the mutation percentage. So basically that the platform forms a concept that digital pathology can be really speed up the process of the conventional tissue biopsy. Smartphone microscopy can also be multimodal. In other words, we can combine different detection modality to the same platform. One of the newest effort in this direction is the demonstration, the AppView platform. So AppView is really a modular smartphone-based devices. It uses a common base, and you can switch different imaging heads to enable different imaging modality, such as bright field and then epifluorescence. And then the benefit for this one is we can really further reduce the cost of the device. So you only need to switch out a small set of components instead of the whole image device. And then for this device, again, we demonstrate one application towards a human health monitoring is the detection of breast cancer in the resource-limited settings. So typically for the breast cancer detection, we still have to go to the conventional pathology like analysis, biopsy analysis. But in the resource-limited settings, because of limited infrastructure, typically the way is using a fine needle to aspirate a few cell samples and then try to make a decision. And what the smartphone can do is using the bright field imaging modality, we can visualize the cell morphology, for example, the nuclei, try to differentiate whether it's a cancer or healthy cells. The fluorescence modality can be used to quantify a rapid molecular assay technology developed by our collaborator at the Duke. Basically, it's a washing-free chip-based immunosandwich assay that can be used to quantify a few breast cancer-related biomarkers. So initial demonstration, in this initial demonstration, we're using the default assay to detect the HER2 biomarkers, and currently we are working on a multiplexing default assay that can be used to detect several other different markers. So really the combination, the chip-based assay and the smartphone, and then we demonstrate the system provides equivalent detection sensitivity compared with a benchtop slide scanner or ELISA assay. One final example for the smartphone-based diagnostics is not only for the human detection, but it also can be used for plant disease monitoring. For plant pathogen detection, one challenge is a plant cell is much more robust and stronger than compared with the human mammalian cells. So cell lysis and sampling is the major challenge to perform a rapid field detection. And in order to overcome that limitations, our solution is to use a low-cost microneedle device made of environmentally friendly polymers such as a PVA. Really, the way to use the microneedle is you simply press the microneedle onto the plant tissue such as a leaf, and let it stay for a few seconds, and you retract the needle devices. And then after that, you collect the sample solution on the surface needle, and those samples are immediately ready for any downstream analysis, such as PCA and many others. So literally, our methods is washing-free and purification-free. And then after the sample extraction, you can deposit the sample into a smartphone-based amplification devices you have seen on the slides. It comes with a sample cartridge to try to receive the sample. The sample cartridge also has a integrated heating element for us to do isosomal nucleic acid amplification. In this case, we implement a multiplexer lamp reaction, so you can run 4 different lamp reactions on the same chip devices for detecting multiple pathogens simultaneously. And then we demonstrate that these smartphone-based amplification devices, they provide quite comparable detection sensitivity compared with the benchtop thermal cycler. And then now we move to the CRISPR technologies, and I think that the audience here is quite familiar with what is CRISPR is. Essentially it is initially a gene editing tools, but in recent years, many other research groups has been converting the CRISPR technology into a molecular detection platform, and then many different detection modality have been proposed. For example, you can do fluorescent CRISPR assay, electrochemical detection, colorimetric fluorescent CRISPR assays. But nevertheless, almost all of the current CRISPR detection assays are based on very simple principle, which is leveraging the unique transcleavage over certain CRISPR enzymes such as Cas12a and Cas13a, try to transcleave a reporter molecules, and then those reporter molecules typically consist of single-stranded sequences to generate a signal that is proportional to the target concentration. So it's all based on that principle, transcleavage of single-stranded reporters. And then one question we have is always, can CRISPR-Cas system transcleave double-stranded reporters? So in the literature, we usually consider that's impossible, and then we tested it by ourselves, and it also confirms, yes, it's what you can see the gel image data is we're using a 1 kb long double-stranded DNA reporter as a substrate for the CRISPR Cas12a system, and we do not see any transcleavage, which agree with the literature reports. However, if you put in a M13 single-strand DNA, they can be transcleaved, and then you see the disappearing the bands. But recently, we found a very interesting family of double-stranded DNA, which is the lambda DNA. We found that although lambda DNA is double-stranded and very long, very stable, they, however, can be transcleaved by the Cas12 system. Very interesting. Initially, we pre-treated the lambda DNA with a slightly higher temperature, 95 degrees Celsius, so on and so forth. We try to set apart, denature the lambda DNA a little bit so that they can be transcleaved. But eventually, we found out those temperature pre-treatment is not necessary. Even under lower temperatures, such as 37 or even room temperature, we still observe the lambda DNA can be transcleaved by Cas12a, and then you clearly see the disappearing the bands when you increase the target concentration. So, and then we try to ask why. This is really contradictory to the conventional knowledge. And then we went to some literature, and some of the literature study suggests there may be a single-stranded region, for example, those loop structure, D loop, or small loop structure along the lambda DNA backbone. So we try to test that hypothesis, see whether it's true. So in order to do that, we design a few synthetic short double-stranded DNA reporter molecules, some with the loop structure, some with do not have the loop structures. And also we change the size of loops, small loop and then big loop. And the results is interesting and also convincing. And then without the loop structure, if you have the brown end double-stranded, again, no transcleavage, the band still there. However, after the introduction of the loop structure, we notice the reporter molecules start to be susceptible to the transcleavage, and then the band start to disappear. So that sort of gave us a hint, and maybe the single-stranded the loop structure could be the initial binding and the recognition site for the Cas12a. And then, of course, there's more studies going on trying to confirm that the potential mechanism. But nevertheless, we're utilizing the very interesting chance cleavage behavior of the lambda DNA, try to come up a new CRISPR-based detection assay, different from all the previous mentioned detection modality, either fluorescent, colorimetric, electrochemical. They always require a labeling. Right? They require label the important molecules with signaling molecules. So that not only increases the cost of the reported molecules, but also increase the cost of the detection devices. So you have to have a sophisticated optical or electrical detection systems. And here what we try to propose a new CRISPR detection assay may have a more point-of-care application potentials. So this methodology is based on the detection of the targets by measuring the size change of the reported molecules. So since lambda DNA can be transcleaved in the presence of the target, and we notice the degradation, or in other words, the size of the fragments of the lambda DNA is inverse proportional to the target concentration. Higher target concentration induces more chance cleavage, and then smaller lambda DNA fragments. And using a very simple gel electrophoresis, we can quantify the target concentration without any fluorescent labeling or optical detection, simply by doing gel electrophoresis. And then we demonstrate that by monitoring the size change. Actually, this mechanism can provide very sensitive detection down to hundreds of femtomole. That's typically orders of magnitude even better than the conventional fluorescent-based reporting system. And then another question we have, is the single-stranded loop structure the only structure that can promote the chance cleavage of the Cas12 system? And then we look at the other structure, for example, we come up with this very interesting overhang structure, which means, based on the initial double-stranded DNA backbone, we add a single-stranded toll hole. And then we noticed after adding the toll hole, the reported molecules becomes, sensitive to the chance cleavage, and then they can be transcleaved by the Cas12a. No single-strand toll hole again, no degradation, no gel band change. And we also look at how long the toll hole need to be, right? So typically we notice we need at least 4 to 8 nucleotides in terms of the length of the toll hole to initiate the chance cleavage. And then we assume that the size of the toll hole is compared to the size of the Cas12 protein. Again, that's an indication. The single-stranded overhang is the initial seeding or anchoring point for the Cas enzyme. More interestingly, the toll hole location is very critical. We notice only if we add the overhang to the 3' end, it can promote chance cleavage. If you add the overhang, single-strand overhang to the 5' overhang, 5' ends, there is still no chance cleavage. So this overhang preference, from 5' over from 3' over 5' is a very interesting. And then we are currently considering this may be related to the very unique direction movement of the enzyme molecules along the DNA backbone. And then the kinetics data also describe the difference between the reaction rates of 5' overhang versus the 3'. Again, the 3', the reaction performs much faster than the 5' is kind of slow. So, yeah, again, we're still undergoing research trying to answer the fundamental question why there's a preference. And one hypothesis here we illustrate in the cartoon is we believe that the Cas12a, like many other enzymes such as polymerase, when they move along the DNA, they have very unique directional behavior. However, which protein subunit in charge of those movements is still under investigation. So using these hybrid reporter molecules, basically the hybrid reporter molecule is referring to a initially double-stranded DNA backbone, but with a short 3' overhang. And then these reporter molecules could be also used a very interesting and a new reporting system for the Cas12a detection system. And then we demonstrate using these reporter molecules for detecting a few real-life model diseases such as AAV detection, HPV detection, so on and so forth. Again, those detection reporter molecules, they do not need any fluorescence labeling. They are very easy to design, low cost, and then they provide very fast reaction kinetics. And then more important, they provide very good detection sensitivities comparable to the conventional fluorescence based reporters, or even better than the sensitivity of the FQ reporters. So for the final few slides, I do want to show how we can combine the two technology together, the smartphone-based devices and then the CRISPR detection. In one of the early studies, we demonstrate a smartphone-based CRISPR assay for COVID detection at the beginning of the pandemic. So basically, it's RPA and CRISPR one-part reaction that can quantify the COVID RNA in 10 to 15 minutes of the reaction time, and then we miniaturize the assay in a disposable and miniaturize the chip. And the chip eventually can be inserted into the smartphone reader devices for the rapid quantification. And then in addition to the detection system, I think another interesting aspect for this work is the demonstration of accurate CRISPR, accurate COVID detection can be done from human saliva samples. This is one of the a few early examples to demonstrate saliva could be a very reliable matrix in addition to the nasal swab for the COVID detection. And then later we demonstrate, one challenge for the CRISPR detection is always the limited detection sensitivity. And one way to overcome that is try to perform the CRISPR reaction in a digital assay format. So digital assay based detection referring to compartment the bulk reaction into small reaction chambers, so that statistically you hope each micro reaction chamber can perform a single molecule detection. And then we demonstrate that by miniaturizing the CRISPR reaction in a micro well chip based array. And then again, using a smartphone camera to quantify the chip, and then you will see essentially we quantify the target concentration by counting how many positive wells instead of measuring the absolute intensity. And then we demonstrate the digital assay is very robust. It can be used for clean buffer solution or could be used for more complicated serine samples. For each sample matrix, we are able to detect the synthetic target down to phantom water level. That's again, typically the bulk CRISPR-based assay, the limit detection is typical on the order of the picomolar. So the digital assay chip can enhance the detection sensitivity at least by several orders of magnitude. The final slide I want to share is going back to the new reporter molecules we demonstrated before, either the lambda DNA reporter or the double-stranded DNA. One challenge currently to translate that technology to a point-of-care detection is the gel electrophoresis typically is still considered a laboratory-based quantification technology, require a stable power sources and a big equipment such as the gel electrophoresis device, the gel imager, so on and so forth. And recently we are putting some effort trying to miniaturize the gel electrophoresis system, again coupled with the smartphone-based sensors, so we can run a mini gel in a smartphone-based portable systems and use a smartphone camera to capture the gel bank shifting. So The system is still under development, and hopefully we're gonna able to present some results in the near future. So really try to summarize. We demonstrate the smartphone can be a transformative imaging devices, can provide sub-micron spatial resolution, single molecule detection sensitivity, and a very tunable field of view from millimeter squares to centimeter squares. And we demonstrate the CRISPR technology has a unique advantage for the point detection because it's a very simple assay design in a one CRISPR enzyme, one guide RNA, and then it's isosomal conditions, and then more importantly, CRISPR can detect very short nucleic acid sequences, both DNA and RNA. So we see that that's why the CRISPR has a tremendous potential as point-of-care technology to replace the traditional PCR-based methods. And the final message is combining the CRISPR diagnostic molecular assay with the smartphone devices really help to complete the loop to form a standalone and then sample to answer quantification system. So in other words, the system not only be able to perform the molecular reaction, but also you have the real devices immediately you can quantify the results, and even utilizing the connectivity of the smartphone to share and process the data and send to your remote doctors or collaborators. So with that being said, I'll conclude here, and then would like to acknowledge all the collaborators, the students in my lab doing the work, and then also the funding agencies. So we don't have a live Q&A sections, but if you have any questions, feel free to contact me by email or leave the question in the Q&A box. Thank you again.